Hi, everyone. My name is Dr. Noelle Cutter. I'm an associate professor at Malloy College in Rockville Center, New York. And I am going to talk to you a little bit today about some of my research based around medulloblastoma, which is a rare but aggressive form of brain cancer found predominantly in pediatric patients. I'm going to wrap up my talk and just speak a little bit about the impact that COVID-19 has had on the diagnostics across the globe, and then as well as look at a way of having a more global response for future outlook within the disease. So medulloblastoma is a devastating illness and is the most common pediatric brain tumor. It is predominantly treated with cytotoxic and non-targeted approaches. Survivors of medulloblastoma suffer from severe treatment-related effects from the radiation and chemotherapy, as well as mortality in general tends to be pretty high. Thankfully, over the last several years, remarkable technological breakthroughs have facilitated the molecular characterization of multiple tumor types and provided new insights into the genetic basis of these cancers, and has prompted innovative strategies that are really changing the management paradigm in pediatric neuro-oncology, not just medulloblastoma. Genomic tests have begun to affect medical decision-making, and in a number of ways, we're starting to understand a lot about the histopathology between the different tumor types, and then develop these novel therapeutic agents for our patients with high risk or poor prognosis tumors. Or alternatively, we can reduce treatment intensity for those that have already a favorable prognosis. However, the impact of COVID-19 the impact that COVID-19 may have on this pediatric population and the management of children with cancer in general remains to be unclear and is poorly documented. So this talk, I hope to examine some recent scientific progress in medulloblastoma research with a focus on the genes, the genetics, the genomic pathways, all these different pathways that are going to drive tumorigenesis in the advancement of conventional and targeted therapy. I'm going to talk to you a little bit about the identification of subgroup-specific regulations and subgroup-specific advances that have been made over the years, as well as a little bit of a discussion on actionable therapeutic targets that may have the potential to revolutionize therapy for patients with medulloblastoma. And overall, the goal is hopefully to significantly enrich overall survival for these patients. So just a little bit about the history. Initial descriptions of medulloblastoma do date back to Harvey Cushing with Cushing's syndrome, who initially described medulloblastoma as a subset of gliomas. He detailed many of the key features of these tumors, including their tendency to arise from the cerebellar vermis and to exhibit leptomeningeal metastasis. Along with neuropathologist Percival Bailey, Cushing described medulloblastoma histopathologically as containing numerous mitoses, small round nuclei, and minimal cytoplasm. So this is what the cells would have looked like underneath the microscope. The two doctors also recognized that medulloblastoma cells resembled undifferentiated cells present in the embryonal stages, and posited that medulloblastoma arose from medulloblasts, which are a type of multipotent stem cell thought to be present in the neural tube. While no such single primitive cell type exists, Percival as well as Cushing, their foundational work has really defined some of the clinical characteristics, operative techniques, as well as histopathology of this tumor, and has really formed our basic understanding of medulloblastoma, which has now been driven by this new molecular charge and the new molecular era. While some early medulloblastoma patients were treated with radiation sporadically, the first landmark change in the management of these tumors came back in about 1953, when a study of 27 patients with medulloblastoma demonstrated significantly improved survival after receiving cranial spinal radiation. So now this routine cranial spinal radiation has improved survival by approximately 60%, which is awesome. The next intriguing progression in our understanding of this disease occurred in the 1970s in a study in which the authors proposed that medulloblastoma can be classified as a primitive neuroectodermal tumor, or PNET, because of its histological similarities and the hypothesis that medulloblastomas and other PNETs arise from undifferentiated cells in the subepidermal zone. Finally, the addition of cytotoxic chemotherapy regimes as a standard of care started with trials in about the 1970s and has greatly improved the 5-year survival risk for the average disease found in patients, although high risk still remains a challenge. Rapid advances now, coming into the 2000s era, rapid advances in molecular genetics over the past 2 decades has really provided significant advancements in our understanding of medulloblastoma. This tumor has entered that molecular era, beginning with gene expression array studies, demonstrating that medulloblastoma is really a distinct entity from other embryonal central nervous system tumors. So our understanding of how we treat it now also has to be based off of these new findings. Multiple independent groups performed transcriptional profiling on medulloblastoma samples, and we've now reached a consensus of 4 distinct molecular subgroups: wingless, Wnt, sonic hedgehog, and then group 3 and group 4, which are a little bit unclear, more so in their classification status. Molecular subgrouping has already influenced the design of contemporary clinical trials, and we'll talk a little bit about that, and has refined preclinical studies of medulloblastoma. And these subgroups were now adopted into the 2016 WHO classification of the tumor within the central nervous system. The expanding wealth of molecular data that has been driven through the studies in the early 2000s has brought us now into where we are today, and our understanding of that molecular data from these patient samples has really started to allow for further refinement of these subgroups, with the numerous subtypes of medulloblastoma now being recognized. So if we want to talk a little bit more about the medulloblastoma molecular subgroups, medulloblastoma, histopathologically is classically considered that round blue cell tumor of the cerebellum, and it occurs exclusively in the posterior fossa and has the potential for leptomeningeal carcinomas. And that's part of what Percival and Cushing had described to us early on in the progression of our understanding of medulloblastoma. Medulloblastoma is the most common malignant pediatric brain tumor and is one of the leading causes of pediatric tumor-related death, and is typically classified as a stage 4 tumor. It's a rapidly growing tumor of the cerebellum, which is the lower rear portion of the brain, and it controls things like balance, posture, complex motor functions. And so tumors located within the cerebellum are also called infratentorial, or the posterior fossa, meaning it is located in this specific region. It has a thick membrane that separates the larger cerebral hemisphere of the brain from the cerebellum, and is found near the brainstem and the cerebellum. Medulloblastoma is the most common of the embryonal tumors, and these tumors arrive from some sort of embryonic or immature cells at the earliest stages of cell development, causing them to be extremely aggressive. They can easily metastasize through the location of the cerebral spinal fluid, and it can then travel to the spinal cord. Symptoms of medulloblastoma depend really on the size and location within the patient, as well as the stage of development. They can start as flu-like symptoms, non-specific symptoms such as headaches, nausea, vomiting, dizziness, loss of balance, vision loss, and if spread to the spinal cord, back pain, and walking problems. Doctors trying to understand patients and what's going on with their patients usually go through common health histories, perform neurological exams, measure brain function, balance, coordination. In addition, imaging tests such as MRI are done to evaluate medulloblastoma. A lumbar puncture can also be done to determine if the cancer cells are found within the cerebrospinal fluid. Current treatments are lacking, but the ones that we do have tend to be of the older following of cancer treatments. So they include surgical removal of the tumor. Surgical removal does allow for the classification of the type of medulloblastoma by its molecular characteristics, as well as treatment with radiation and treatment with chemotherapy. However, these types of therapies have really not greatly increased 5-year survival rates since the 1970s. So in general, we seem to be kind of stagnant at that 70 to 80% for our standard risk patients and 60 to 65% for our high risk metastatic patients. There's also a dismal prognosis for some medulloblastoma patients, with 5-year survival windows only being about 30 to 50%. This slide here shows you the breakdown of the molecular subgroups. As you can see, several pathways have been implicated in medulloblastoma formation, and knowledge of these pathways is now being used to develop new ways of treating children with medulloblastoma. During the past decades, research studies have greatly improved our understanding of medulloblastoma oncogenesis through elucidation of several developmental signaling pathways, namely those that we've just spoke about, the sonic hedgehog and the wingless Wnt and Notch pathways. These pathways involve cell signaling receptors, intracellular second messengers, transcription factors, and gene regulation. Tight regulation of these signaling cascades is essential to normal cerebellum development, and therefore dysregulation of these pathways has been clearly linked to medulloblastoma tumorigenesis. Furthermore, the surge of advancement in genome analysis technologies has really accelerated our understanding of the molecular basis of medulloblastoma. Medulloblastoma is no longer considered a single disease, but rather comprised of at least 4 molecular distinct subgroups, each of them characterized by discrete clinical presentation, demographic features, prognosis, expression profiling, and genomic abnormalities. The identification of molecular subgroups has a great impact on clinical management, including patient stratification, treatment strategy, and the design and implementation of targeted therapy. The 4 molecular subgroups are created based on genomic characterization that was described using expression profiling, whole genome sequencing, whole exome sequencing, as well as methylation profiling. A lot of my specific research is dedicated to understanding the G3 subgroup, or the group 3, mostly because very little is known about this subgroup. So for example, group 3 has a very aggressive metastatic disease and is very common in the youngest population of our medulloblastoma children. And so a lot of the work that I focus on looks at the role that immunotherapy plays as a treatment option for patients that are diagnosed with group 3 medulloblastoma. The molecular subgroups of medulloblastoma have some consensus profiles. So this current consensus agrees upon these four distinct molecular entities of medulloblastoma that is characterized by specific mutations, copy number alterations, transcriptomic and methylomic profiles, as well as clinical outcomes. Subgroup assignment is prognostic with markedly different survival rates. And we usually use that 5-year overall survival as being our marker. So overall survival is as high as 95% in the WNT subgroup, while group 3, which is the group that I focus on, patients seem to fare the worst and with the shortest survival and also the youngest classification in patients. So it's usually diagnosed in infants, for example. And intermediate, about 75 to 80% overall survival, characterizes the group 4 and the sonic hedgehog medulloblastoma, although it really depends on histology, presence of metastasis, as well as molecular abnormalities such as mutations and oncogene amplifications. Only the most frequently altered genes are listed in group 3 and 4 on this slide, although there's many transcriptomic and methylomic profiles and mutation sequences that you can pull up any of the references to get more available information on that group specifically. Adult samples for medulloblastoma tend to be extremely rare and specifically rare for group 3 patients, while the majority of group 4 tumors consist of children as well. So we're going to go through a little bit of background on some of the different molecular subgroups to give them a little bit more contextuality and some more definitions and descriptions. So we'll start with the WNT signaling pathway for medulloblastoma. So biologically, the WNT subgroup of medulloblastoma is characterized by mutations, as you would imagine, in the WNT signaling cascade, and subsequent aberration activation of the canonical WNT signaling pathway. Germline mutations of the WNT pathway inhibitor of APC may predispose patients to tumorigenesis and may increase their susceptibility to several other cancers, including medulloblastoma. Somatic mutations found in the WNT subgroup include the CTNNB1 gene encoding beta-catenin, and tumors show a monosomy on chromosome 6, DDX3X mutations, and TP53 mutations. A potential choice for targeted therapy is WNT inhibitors, of which several are currently in early phase 1, phase 2 clinical trials for patients who have this WNT-activated solid tumor within the WNT subgroup for medulloblastoma. In general, they do tend to have an excellent survival rate, close to 95%, when treated with conventional means. Sonic hedgehog signaling in medulloblastoma tends to be disrupted along its signaling pathway and plays an important role in the pathology of this disease. The first indication that mutations in the sonic hedgehog pathway contribute to medulloblastoma was a finding that Gorlin syndrome patients who inherit germline mutations in the patch gene are predisposed to medulloblastoma. Patched1, for example, is a transmembrane protein that functions as a receptor for sonic hedgehog. In the absence of sonic hedgehog, Patched1 maintains the sonic hedgehog pathway in the off state by acting on smoothin. Upon sonic hedgehog binding to Patched1, this repression is lifted and a signal is transduced through the smoothin to the nucleus, and that increases expression of target genes, including GLI1 and Patched1 itself. This pathway is crucial for normal development of the cerebellum because it governs the proliferation of the granular neuron precursor cells, the most abundant neuronal population in the brain. The granular neuron precursor cells are believed to be the cells of origin for medulloblastoma, although we don't really have definitive proof here. So the research is still out on that understanding. It has been suggested that medulloblastoma arises from an aberration of normal developmental processes, and that these granular neuron precursor cells fail to exit the cell cycle at their appropriate time, and therefore remains in the EGL, eventually expanding to form a tumor. This finding that over 30% of sporadic medulloblastoma show elevated expression of GLI1 does support this hypothesis. So in terms of the sonic hedgehog subgroup, we often see somatic mutation in genes along the pathway, which include patched, smoothen, SU-FU, in addition to amplifications of that GLI1 and GLI2, as well as MYC. The sonic hedgehog subgroup is currently one of the better studied of the medulloblastoma subgroups. We can look at things like the receptors such as patched. We look at the SU-FU, smoothened. Collectively, this is found in about 10 to 15% of the cases of sporadic medulloblastoma that lead to the expression of the oncogenic transcription factor GLI1. In vitro as well as in vivo studies suggest that the sonic hedgehog pathway inhibitors can be used as therapeutic agents for this medulloblastoma subgroup. And so there's been a lot of studies that have looked at several different types of inhibitors, some mechanisms that affect smoothen inhibition. In addition, one therapeutic approach is using the smoothen inhibition has been shown to be quite effective. And a more recent therapeutic approach may look at some of the epigenetic treatments for the bromodomain inhibitors. So the bromodomain inhibitors have been shown in both in vitro as well as in vivo studies to decrease cell viability and proliferation of the sonic hedgehog medulloblastoma subgroups. But we really don't have any good clinical data because no clinical trials have yet been examined looking at these bromodomain inhibitors as potential therapeutics from this medulloblastoma subgroup. There's also other approaches that may use MET inhibitors that have been shown to decrease tumor cell proliferation, as well as induce apoptosis in both in vitro and in vivo approaches, which again also provides a strong rationale for some sort of clinical evaluation. Moreover, for this subgroup of patients, a clinical trial is ongoing to evaluate the doublet therapy comprising a cyclin-dependent kinase, CDK4/6 inhibitor with either gemcitabine or trametinib or songebic in adults with refractory or recurrent sonic hedgehog medulloblastoma. Alternative sonic hedgehog inhibitors may constitute other sort of treatments that have helped to lead to increased degradation of the GLI transcription factors. So these various treatments may be particularly suitable in tumors that have developed resistance to maybe some of those earlier previous treatments, such as the smoothen antagonists, or as secondary resistance that was mainly caused by GLI2 amplifications in preclinical models. In addition, the phospholinositol 3 kinase, protein kinase B, mammalian target rapamycin, or the mTOR or protein kinase A signaling inhibitors are also a potential interest for signaling cascades that are targeted by mutations and result in this GLI activation. Drugs targeting the TP53 pathway, such as Nutlin-1, allow the wild type copy of the TP53 to become active and thus negating the deleterious effects of a dominant negative TP53 mutation, which is found in about 13% or so of sonic hedgehog tumors. However, current efforts are focused on proper patient selection in trial design, of any of these therapies that may be viable for future clinical trials. Group 3 medulloblastoma is a lot of what my research is focused on. So group 3 medulloblastoma patients do have the worst clinical outcome, with 5-year survivals approximately about 50%, along with high rate of metastatic dissemination at diagnosis. Low proportion of recurrent single nucleotide variant makes this group remarkable and can be considered as a copy number driven subgroup. This subgroup is characterized by several amplifications resulting in high MYC mRNA expression levels compared with other groups such as group 4 and sonic hedgehog tumors. In addition, transcriptional profiling has revealed that the retinal gene signature in G3 tumors, and due to the poor outcome of patients with G3 tumors, there is an imperative need for novel therapies, specifically in those patients that harbor these high levels of MYC amplifications. Indeed, the MYC amplification is the most common somatic copy number alteration that has been identified in this subgroup and is commonly associated with a fusion of the non-coding exon 1 of PVT1 and exon 2 of MYC. The idea of inhibition of MYC is an interesting, but yet very challenging therapeutic strategy for this subgroup. Several small molecular inhibitors of MYC have been proposed, and some are in early clinical trials. An added possible strategy to target MYC might be to inhibit some downstream effectors of MYC, specifically the bromodomain proteins. And this strategy has been seen to be successful in some preclinical models of various MYC-driven malignancies. In addition, the transforming growth factor beta, the TGF beta signaling pathway, is also upregulated in approximately 20% of the cases, as evidenced by somatic copy number aberrations. And currently, there are several trials of TGF beta inhibition for a multitude of cancers. Isochromosome 17q constitutes the most frequent broad genomic imbalance, and there are certain actionable targets driven by this alteration that remain to be identified. So there's still a lot that we don't know about this subgroup. In certain murine models of group 3 medulloblastoma that do exist, suggest that it is driven by this overexpression of MYC and inactivation of TP53 in the CD133 neural stem cells of the postnatal cerebellum, which can be possibly used for the design of preclinical trials in the future. The group 4 of medulloblastomas is the most prevalent subgroup with a high male and female ratio that has the propensity for metastatic dissemination at diagnosis and an intermediate prognosis with a 5-year overall survival rate at approximately 75 to 80%. The cell of origin is unknown for this subgroup, and currently a possible preclinical model for group 4 medulloblastoma exists with a MYC-N overexpression under the GLT1 promoter. Genomically speaking, group 4 is characterized by a high incidence of isochromosome 17q and recurrent tandem duplications of the Parkinson-related gene SNCAIP and the MYC-N amplification. Single nucleotide variation in certain histone modifiers are also enriched in this subgroup, and specifically mutations in H3K27, the ME3 demethylase, and KDM6A occur in approximately 13% of group 4, and mutations in the H3 methyltransferase MLL3 occurs in about 5% of the population. As such, the idea of epigenetic modifying drugs may be a very attractive therapeutic strategy in a subset of patients with group 4. Deletions that affect NF-κB1A, as well as USP4, and regulators of the nuclear factor kappa B, NF-kappa B pathway are enriched in group 4 patients, making NF-kappa B a possible rational therapeutic target for group 4 medulloblastomas. As with inhibition of downstream effectors of MYC in group 3, inhibition of downstream effectors of MYC-N is a possible therapeutic target. And recently it has been shown that bromodomain inhibition in MYC-N-driven neuroblastoma is an effective in preclinical models. Identification of a subset of group 4 with poor prognosis may help with the identification of personalizing therapy, whereby escalation of current therapies can be considered in high-risk cases. Conversely, delineation of patients with good prognosis in spite of metastatic dissemination these patients would be ideal candidates for therapy de-escalation, specifically as these patients currently receive high doses of craniospinal irradiation. Mutations can comprehend in genetic predisposition to the development of medulloblastoma through defects in pathways important in their pathogenesis. Therefore, it is very fundamental that we understand the pathogenesis that is underlying for medulloblastoma patients. So now I'm going to talk a little bit about the role that some of these epigenomic studies have resulted in our understanding of medulloblastoma. So numerous epigenetic alterations exist across all medulloblastoma subgroups, specifically though in group 3 and 4. Epigenetic alterations are of immense interest right now, as several approved drugs target these epigenetic changes, specifically the histone deacetylase inhibitors, and for several other brain tumors. Epigenetics is the heritable changes that do not involve perturbations in the nucleotide sequences. And so therefore, these modifications are mediated by the things such as DNA methylation, histone modifications, chromatin remodeling, microRNAs, and long non-coding RNAs. DNA methylation is one of the more well-characterized epigenetic phenomenon, which typically uses sites at CPG islands in the promoter regions of genes that results in transcriptional repression. However, it is unclear at the present time how targeting DNA methylation as a therapeutic strategy is going to be helpful in medulloblastoma patients. Histone modifications, which include things like ubiquitination, acetylation, phosphorylation, and sumylation, are modified by epigenetic modulators. Non-coding RNAs, which include microRNAs and long-coding RNAs, regulate gene expression, typically by gene silencing, at the transcriptional or post-transcriptional level. Epigenetic changes occur spatially and temporally throughout development, and they may influence processes such as DNA repair, cell cycle, and cellular differentiation. Epigenetic regulation plays an important role in cancer, often resulting in the silencing of tumor suppressor genes or the activation of oncogenes. Therefore, the role of epigenetic modifications in disease pathogenesis could be an emerging focus for cancer therapeutics. The DNA methylation and histone modification patterns associated with the development and progression of cancer really do have potential clinical use. DNA hypermethylation markers are under study as complementary diagnostic tools, prognostic factors, and predictors of response to treatment. Unlike mutations, DNA methylation and histone modifications are reversible. So epigenetic alterations allow the cancer cell to adapt to changes in their microenvironment, but dormant, hypermethylated tumor suppressor genes can be awakened with drugs. So it is possible then to re-express DNA methylated genes in cancer cell lines by using demethylating agents and to rescue their functionality. So DNA methylating drugs in low doses have clinical activity against some tumors. And right now, we are currently looking into this practice in our lab, but more work is going to be needed to test their efficacy in solid tumors, such as medulloblastoma. As far as treatment options in medulloblastoma, we really haven't progressed much since, the 1990s and the early 2000s. So treatment usually involves first tumor resection. The importance of tumor resection was recognized by Cushing back in the early days of medulloblastoma work, who noted an increase in survival in patients who had undergone radical resection versus biopsies alone. The importance of the extent of resection has been revisited in observational studies over time, really refining our understanding of how best to surgically manage these patients. These studies have generally demonstrated how best to demonstrated that gross total resection have similar outcomes, suggesting a general guiding principle of the maximal safe resection. Recently, the benefit of extent of resection was reanalyzed while controlling for the molecular subgroups in a cohort of about 787 patients. After controlling for subgroup across the entire cohort, there was a progression-free survival benefit with gross total resection over that with subtotal resection, but no benefit for progression-free survival or overall survival when comparing the gross total resection to the end total resection, the residual tumor, the total amount of the residual tumor that was left. In the analysis of the progression-free survival and overall survival, or for the individual subgroups, only the patients that were found to be in group 4 tumors appear to have a PFS benefit with GTR over STR. So to summarize, while GTR still remains the ideal goal of surgery, so gross total resection of the tumor, similar outcomes can be expected in patients with minimal residual tumor, and surgeons therefore must weigh the potential neurologic morbidity of a more complete resection against leaving some residual tumor. Of note, an important goal of surgery, of course, is obtaining sufficient amount of tissue so that you can run histopathological diagnostics as well as molecular analysis. Given the spatial molecular heterogeneity within the primary tumor, it is also important to take multi-regional tissue sampling during resection, especially in regions of residual tumors, to determine the ubiquity of the genetic and epigenetic alterations, as well as aiding in the rational selection of targeted therapy. The role of resection for locally recurrent medulloblastoma has not really been rigorously investigated, but studies have suggested a benefit of repeated resection. However, these studies tend to have highly selected patients, and analysis accounting for molecular subgroups are lacking. Nonetheless, given the divergent evolution and clonal selection that occur throughout therapy, biopsy or repeat resection of recurrent tumor may provide good molecular insight to guide therapy for recurrence. Other treatment options do include radiation therapy. So in children older than 3 to 5 years of age, the current convention for radiotherapy involves radiation to the entire cranial spinal axis in order to treat or provide prophylaxis against metastatic recurrence. Patients without metastatic disease who have had at least some tumor resection of the primary tumor receive anywhere on the order of 23 to 24 gray dose of craniospinal radiation and a 54 gray boost to the tumor bed specifically. Radiation strategy for patients who present with metastatic disease are more variable, with patients in most of North American centers receiving anywhere on the order of about 36 gray dose of craniospinal radiation and a 54 gray dose to the primary tumor bed and a 50 to 54 gray boost in the nodular metastatic deposits. In Europe, however, patients with metastatic disease often receive radiotherapy following chemotherapy in either conventional single dose per day or hyperfractionated 2 doses per day regimens. Current areas of investigation are establishing the efficacy of treatment de-escalation for wet medulloblastoma, as well as incorporating alternative radiation technologies, such as proton beam therapy in place of photon-based radiation. And then finally, our third option for treatment is chemotherapy. So current chemotherapy regimes are primarily determined by the patient's age and a suitability for radiotherapy, as well as risk stratification. So in children over 3 to 5 years old who receive radiation and have had some level of tumor resection and no metastasis, the current gold standard of care proposed by Children's Oncology Group is a chemotherapy regimen starting with weekly vincristine, concurrent with radiotherapy, followed by 8 cycles of cisplatin, vincristine, or either a cyclophosphamide or lomustine. The optimal chemotherapy regime in patients characterized as high risk in the non-molecular risk stratification system has really not been defined. So there is a little bit more variability there in the chosen agents and the number of cycles. For example, a recent Children's Oncology Group trial used carboplatin and vincristine during radiotherapy, followed by 6 cycles of cyclophosphamide and vincristine, with some patients also receiving cisplatin. Other trials have investigated the use of intraventricular methotrexate and high-dose chemotherapy with autologous hematopoietic stem cell transplantation. And in young children, so, if we're talking about pediatric patients under the ages of 3 to 5 years old, of which the neurological impairment caused by radiotherapy may preclude its use, multi-agent chemotherapy is used to really delay the use of radiotherapy. As in older patients with high-risk disease, clinical trials have also incorporated other methotrexate and high-dose myeloablative chemotherapy with autologous hematopoietic stem cell transplantation for young children with medulloblastoma. So those are some newer treatments. And again, it's very specific on status of the disease as well as age of the patient. Targeting medulloblastoma metastasis has always been a challenging feature, but based on the most recent data, if we look at figure A, the primary tumor in the cerebellum is usually composed of distinct subclones, of which select clones may have metastatic potential. In figure B, we look at metastatic clones that may shed from the primary tumor into the cerebral spinal fluid circulation or extravagate into local circulation. In figure C, we're looking at metastatic cells as they transition via the cerebral spinal fluid route or some other blood-based route to colonize in distinct leptomeningeal sites. And then in figure D, metastatic clones undergo further genetic divergence in the leptomeningeal niches and undergo complex interactions within the tumor microenvironment. And this may include things like immune cell interaction, as well as neovascularization. Medulloblastoma metastasis is very common, yet little is known about the molecular programs necessary for medulloblastoma metastasis and survival. And currently there are no therapies that target this paramount clinical problem. Preclinical studies have demonstrated that metastatic medulloblastoma, like recurrent medulloblastoma, do contain distinct molecular events compared to those of the primary tumor and have been identified, and we have identified potential driver pathways such as PI3K signaling pathway. In terms of where we are with emerging therapies, really there is such an increased tendency in oncology in general to explore the efficacy and safety of molecularly targeted agents for sample-specific genetic alterations, irrespective of the tumor or tissue type. The expanding number of new trials investigating immunotherapy really reflects recent tendencies in oncology. Currently, the number of phase 1 studies are really out there to kind of give us an idea of understanding more targeted approaches in medulloblastoma, and they're going to combine our results from genomic, transcriptomic, high throughput drug screening across many diverse molecularly characterized patient-derived models, but really the clinical need for them is so great. Right now, a lot of the work that is coming out of a lot of labs, including my own, is focused on immunotherapy and really expanding the number of clinical trials that are underway. So in terms of where we are with immunotherapy as a possible emerging treatment for medulloblastoma, the first group that we're going to look at is the immune checkpoint inhibitors. So immune checkpoint inhibitors are a recent area of research that we've seen in a lot of other tumors. Cancer cells can upregulate immune checkpoints to escape T-cell mediated immune responses. Groundbreaking application of immune checkpoint inhibitors in numerous solid tumors, including metastatic melanoma and renal cancer, has generated interest in their applicability to other central nervous system tumors. The approach is especially attractive for patients with advanced disease and limited options for therapy. So the success of using immune checkpoint inhibitors is limited so far, but of note, most studies are retrospective and observational with limited sample sizes and unselected patient population. So more work is needed in that field. Other attractive areas of treatment also include oncolytic viruses. So cancer therapy that utilizes oncolytic viruses has several advantages over traditional approaches. Oncolytic viruses replicate selectively only in the cancerous cells, but not in the normal cells. And after infecting a few cells, oncolytic viruses quickly spread through the entire tumor. The anti-tumor response is based on a dual mechanism of action. First, oncolytic viruses induce direct lysis of the tumor cells. And second, as a result, the release of the neoantigens to the tumor microenvironment triggers activation of the immune system, facilitating tumor eradication. Based on promising results from preclinical studies, several types of viruses are being investigated in the treatment of pediatric brain tumors. Another possible therapy is CAR T therapy. In CAR T therapy, genetically engineered T cells expressing artificial chimeric antigen receptors with both antigen binding and T cell activating properties are utilized. CAR T therapy has resulted in breakthroughs in treating a lot of blood cancer malignancies, but solid tumors pose a wide array of challenges, such as finding tumor-associated antigens enriched in tumors, but not expressed in the normal tissues. HER2 is frequently overexpressed in medulloblastomas, and intraventricular administration of autologous HER2 CAR T cells effectively cleared orthotopically implanted medulloblastomas in tumor cells and in preclinical mouse medulloblastoma models. Again, more research is needed there to kind of bring it to the clinical trials that would be absolutely necessary. And then finally, another emerging immunotherapy is through the use of natural killer cells. Natural killer cells play a major role in immune defense against malignant disease by recognizing a broad spectrum of tumor cells without specific antigen identification. Natural killer cells are controlled by both activating and inhibitory receptors, and medulloblastoma cell lines expressing specific ligands that are able to trigger natural killer cell activation, enabling the lysis of medulloblastoma cell lines in vitro, regardless of the presence or absence of the stem cell marker CD133. However, complete elimination is unlikely due to inhibitory signals generated by the tumor cells. More work here is going to be absolutely necessary for future outlook. And so finally, we come to the idea of how has the COVID-19 pandemic impacted medulloblastoma diagnostics, treatment, and where do we go from this point onwards? It is already clear that maintaining services as usual during the COVID-19 pandemic is, in many settings, no longer possible. There is an urgent need for all healthcare providers to evaluate their pathways of care to ensure the continuity of curative treatments and palliative care as effectively as possible. Consideration must be given to how families access care in the presence of serious lockdowns and curfews, and service delivery may need to be adapted to availability of qualified staff and suitable facilities. Moreover, in this time of uncertainty and fear, support for patients and their families at diagnostics and during treatments is crucial to ensure that they can complete treatment safely. Just as importantly, as a professional childcare cancer community, we must advocate on behalf of the healthcare workforce to protect the health and safety of nursing, medical staff, as well as support staff. It is their availability that will ensure children with cancer will survive this pandemic. The true impact of COVID-19 infection on children undergoing cancer treatments is at present unknown. All we can do is recommend the principle that the standards of care for the diagnostics, treatment, and supportive care for children with cancer should not be compromised in any way, shape, or form, or electively modified during this pandemic, if at all possible. Globally, the majority of cancer care services are located within the combined adult and pediatric hospitals, which may present with possible interference on treatment for our pediatric population. So how do we set forth recommendations and support during the pandemic? So here I try to set out reasonable and safe modification to both the diagnostics as well as treatment of children with cancer in centers with significant pandemic-related resource constraints. As there's no evidence base to really inform or guide cancer care during a pandemic, these recommendations are formed collectively from expert opinions and based on the principles of pediatric oncology. Moreover, there's no current evidence to support the elective reduction in cancer treatment to prevent or mitigate COVID-19, but deferring elective high-risk treatments may improve patient safety and preserve service capacity to meet the pandemic demand. I think really what we have to begin to do is weigh the risks and benefits of absolutely necessary treatments as versus the elective treatments that our pediatric patients might need to have. As far as our future outlook and our understanding of medulloblastoma, for modest improvements in outcome and efficacy equivalent studies, sufficient number of patients are required to achieve statistically relevant endpoints. There has been tremendous paradigm shift in our understanding of the pathogenesis as well as the biology of medulloblastoma. The advent of rational treatment strategies has been relatively delayed in medulloblastoma compared to other cancers. However, the recent identification of numerous novel therapeutic targets holds promise for targeted and less toxic therapies. Treatment strategies based on biology and not simply on clinical risk stratifications are starting to emerge, especially in patients with tumors belonging to the Wnt and sonic hedgehog subgroups. There is an urgent clinical need to shift research focus toward the less well understood subtypes, such as the MYC amplified tumors, to understand the driver mutations. Key information is provided by the wealth of genome-based data available and novel preclinical patient-derived tumor models. Recent advances and developments in drug discovery and immunotherapy often offer hope to improve cure rates for patients with medulloblastoma while allowing these children to reach adulthood with increased health span. Prospective trials are needed to confirm the validity of molecularly grounded stratifications, eventually offering more precise treatment strategies. The ongoing transition toward molecularly guided clinical intervention is slow, especially for newly diagnosed medulloblastoma patients. Clinical trials aim to fine-tune invasive radio and chemotherapies to reduce intervention-related morbidity in low and standard risk patients, with a particular focus on young children. By 2030, the number of cancer cases is projected to reach anywhere on the order of 24.6 million, and the number of cancer deaths to 13 million. Worldwide, health systems, especially in low-income and middle-income countries, are very ill-prepared to manage this increasing cancer burden. Globally, there is a shortfall in coverage of cancer service for prevention, screening, treatment, and palliative care. If cost-effective interventions for cancer screening, prevention, and treatment were delivered through strengthened health systems, they could largely avoid many premature deaths, unnecessary suffering, and unacceptable inequalities. Yet these interventions are not reaching those in need, and the opportunity to close the equity divide has not been realized. So I close today and just again put my humanitarian side forward and say that we really are going to need a global response in the effective diagnostics as well as treatment of all cancers, and that the COVID-19 pandemic has certainly thrown a wrench in that plan. But I truly believe that we're coming out of the pandemic, and we'll be able to refocus our health gains and our global approach to curing cancer, and that the development of rational treatment approaches, especially for those very high-risk pediatric patients, those with metastatic disease, is going to really be a priority and change the trajectory of this disease for the future. So with that, I will take any questions that you might have.